Hi class, this is your instructor, Skylar Huff, and welcome back. We're now in the 7th chapter, and this is Energy and Metabolism. All living things require energy to carry out life processes. And keeping that in mind, I would say that's what life is. It's the process by which of getting the energy you need to live, and of course using that energy in, of course, a number of ways. So along the way, it may seem obvious that cells need energy to grow and reproduce, but even non-growing cells need energy simply to maintain themselves. Cells obtain energy in many forms, but that energy can seldom be used directly to power cell processes. For this reason, cells have mechanisms that convert energy from one form to another. The ordered systems of the cell provide information that makes these energy transformations possible, because most components of these energy conversion systems evolved very early in the history of life. Many aspects of energy metabolism tend to be similar in a wide range of organisms. The sun, which I know you all know much about, is an ultimate source of almost all the energy that powers life. It's this radiant energy that flows from the sun as electromagnetic waves. Plants and other photosynthetic organisms capture an approximate 0.02% of the sun's energy that reaches Earth. So as we will get to chapter 9 here shortly, photosynthetic organisms convert radiant energy to chemical energy in the bonds of organic molecules. It's this energy that becomes available to plants and animals, such as the giraffe you'll see on page 150 of your textbook. Other organisms class, they also process this, but through the process of cellular respiration. In respiration, as we'll get to in the 8th chapter, our next chapter, organic molecules are broken apart and their energy is converted to more immediately usable forms. So, as I've stated, such as this, what we're about to get into, class, are those basic principles that govern how cells capture, transfer, store, and use energy. And we'll also discuss the functions of ATP, which is known as adenosine triphosphate, and other molecules used in energy conversions, including those that transfer electrons in oxidation, reduction, or redox reactions, as you all learned about in the 2nd chapter of the text. So we will also pay particular attention to the role of enzymes in energy dynamics by way of cells. So let us begin. This is on page 151, and this begins what is known as biological work. So energy is one of the most important concepts in biology, and it can be understood in the context of matter, which of course is just whatever it is that has mass and takes up space. So keeping that in mind, energy is simply defined as that capacity to do work. So it's that any change in the state or motion of matter. So you could say that mass is technically a form of energy, which is the basis behind the energy generated by the sun and other stars. So it's more than 4 billion kilograms of matter per second that are converted into energy in the sun. So biologists generally express energy in units of work known as kilojoules, as you see indicated here. So it can also be expressed in units of heat or heat energy known as kilocalories, which you all can see here. So with that, it's that thermal heat energy that flows from an object with a higher temperature to an object with a lower temperature. So to help you with it, of course, 1 kilocalorie is equal to 4.184 kilojoules. So let us now continue. Potential energy. So as we are here, there's the picture you see down below with that potential energy, that energy of position, and the kinetic energy, the energy of motion. When an archer draws back a bow, kinetic energy, the energy of motion, is used and work is performed. The resulting tension, however, in the bow and string represents that stored or potential energy, as you see here. Potential energy is the capacity to do work as a result of position or state. So when that string is released, this potential energy is directly converted into kinetic energy, as you all see there. And that's that energy in the motion of the bow which propels the arrow. So most actions of organisms involve a series of energy transformations that occur as kinetic energy is converted from potential energy, or that, of course, as potential energy is converted into kinetic energy. So what's a bit different here is what I'll get to with chemical energy. So chemical energy is that potential energy that's stored in those chemical bonds. So it is of particular importance to organisms. So in our example, the chemical energy of the food molecules is converted to kinetic energy in the muscle cells of that archer, as you saw just moments ago. So the contraction of the archer's muscles, which you all can now see here, to of course draw back that bow with maybe 55 pounds or 50 pounds. And then of course that allowed that archer to pull back the bow by way of the contraction of the archer's muscles, like many activities by an organism, is an example of mechanical energy. And I'm just writing it out now. Mechanical energy. I'm going to abbreviate energy class just by the letter E. And mechanical energy is what performs work by moving matter. So there you have it, class. Now move on to the Laws of Thermodynamics. You'll find this in section 7.2, page 151. Thermodynamics is the study of energy and its transformations. It governs all activities of the universe, from life and death of cells to the life and death of stars. So when considering thermodynamics, scientists use the term system to refer to an object that they are studying, whether a cell or an organism or even, of course, planet Earth. So with this, a closed system does not change energy with its surroundings, as opposed to the open system, which can exchange energy with its surroundings. And to help you out with that class, biological systems are open systems. So let's get those two laws about energy, being those first and second laws of thermodynamics, which apply to class all things in the universe. The first law of thermodynamics. Class, energy cannot be created or destroyed. Period. 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 Energy cannot be created or destroyed. Although it can be transferred, we'll convert it from one form to another, including class, the conversions between matter and energy. So as far as we know, the total mass energy present in the universe when it was formed, approximately 14 billion years ago, equals the amount of energy present in the universe today. So with that, it is just all of the energy that can ever be present in the universe. So I would just say, similarly, the energy of any system plus its surroundings is constant. So with this, just keep in mind that by way of this first law of thermodynamics, organisms cannot create the energy required to live. Instead, they must capture the energy from the environment and transform it to a form that can be used for biological work. Now to that second law of thermodynamics. So here it states that when energy is converted from one form to another, some of that usable energy is going to be, of course, available to do work. And of course, other energy is going to be converted into heat that disperses into its surroundings. And when I get to those surroundings, I like to refer to those surroundings class as being entropy. Meaning that's what happens when, of course, energy is being converted from, of course, this form to that form. It is lost as heat, and it goes, class, to entropy, which we'll get to, class, here shortly. So as we learned, class, back in chapter 2, heat is the kinetic energy of randomly moving particles, as it states right here. So with that, unlike heat energy, which flows from an object with a higher temperature to one with a lower temperature, this random motion cannot perform work. So as a result, the amount of usable energy available to do work in the universe decreases over time. So it is important to understand this second law of thermodynamics, and it is consistent, class, with that first law. That is, the total amount of energy in the universe is not decreasing with time. However, the total amount of energy in the universe that is available to do work, I repeat, that is available to do work, is decreasing over time. So, as I've said this way, keep in mind, this is what I just said. As I've said it this way, that less usable energy is more diffuse and disorganized. That entropy, that is lost, that heat that is lost. So what I'm saying here is entropy is a measure of the disorder, or that randomness. That organized usable energy has low entropy, whereas disorganized energy, such as heat, has high entropy. So entropy, class, is increasing. Increasing. So what I'm getting to here is it may be at some time billions of years from now that all energy will exist as heat uniformly distributed throughout the universe. And of course, if that happens, the universe will cease to operate because no work will be possible. So everything will be at the same temperature. So there will be no way to convert the thermal energy of the universe into usable mechanical energy. Just keep this in mind, please. Next up is Energy and Metabolism, section 7.3. So the chemical reactions that enable an organism to carry on its activities, meaning to grow, to move, maintain, and even repair itself, including the ability to reproduce and respond to stimuli, together make up its metabolism. So you can say it's that sum. You can say it's that sum of all the chemical reactions that take place in that organism, to be meaning what the metabolism is. So the sum, that's what I just wrote for the sum symbol. So from chapter 1, you learned about metabolism being that sum of all chemical activities taking place in an organism. So an organism's metabolism consists of many intersecting series of chemical reactions or pathways. Two main types of metabolism are anabolism and catabolism. This should ring a bell. So those anabolic reactions, or anabolism, includes, of course, the various pathways in which complex molecules are synthesized from simpler substances, such as the linking of two small amino acids to form those colossal polypeptides known as proteins. On the converse, catabolism, or those catabolic reactions, include the pathways in which larger molecules are broken down into their component molecules, their smaller ones, such as the degradation of starch. Yes, I did say starch, and I hope you all remember, starch, is it a monosaccharide, a disaccharide, or a polysaccharide? Starch. So as starch is broken down to form monosaccharides, which I gave you the answer, what happens, of course, is it's a catabolic reaction. So, of course, energy will be liberated. So, as you will see, these changes involve not only alterations in the arrangement of atoms, but also various energy transformations. So, catabolism and anabolism are complementary processes. The catabolic pathways involve an overall release of energy, which I just stated, and some of which, of course, power analog pathways, which have an overall energy requirement. So let's get now, class, to where we're going with what's next. Entropy. So when you think of entropy, it's just the equivalent to being that total bond energy. And you'll see entropy, is just being, of course, by that specific type of chemical bond that has a certain bond energy defined as the energy required to break that bond. Up next is free energy. So free energy is what does work. And a lot of times you may hear it called Gibbs free energy or being delta G, which we'll get to class in just a moment. But this is that amount of energy available to do work under conditions of a biochemical reaction. So it's also, as mentioned, known as Gibbs free energy. And this was named for J.W. Gibbs, a Yale professor, who was one of the founders of the science of thermodynamics. So now you see the forming class delta G. That's, of course, free energy or Gibbs free energy. So the change in, of course, H minus temperature times the change in H. Make it make sense. So what we have here, class, is that H represents enthalpy. That H represents enthalpy. I don't know why my line on my T went that way. And, of course, the G represents free energy. I've said it once, but I guess I should add it now. That represents free energy. And I'll just write free E for free energy. And with that, T is that temperature, as I mentioned just moments ago. T is the temperature. And the S, as in Sam, class, is entropy. So there you have it. So this is that equation that can be used to determine, of course, the amount of free energy that is there and available to do work. So in an exergonic reaction, it's a release of energy and is said to be a spontaneous or downhill reaction from that higher to lower energy. And it's because the total free energy in its state is less than the total free energy in its initial state. In other words, class, delta G, meaning if there is energy that is available, it, class, will be negative. Delta G will be a negative number. And so, getting on to the next. Endergonic reactions, on the converse, do not have, class, energy that is there and available to do work. Meaning, there's a gain of free energy, and because the free energy is a product is greater than the free energy of the reactions, delta G has a positive value. So, in this case, there is, class, no energy that is there and available to do work. So, looking here closely, it mentions free energy increases during an endergonic reaction. So if you look closely, endergonic reaction, energy class that is free is increasing here. Over time, you go from reactant class to product, you get that increase in free energy. On the converse, of course, you saw this, that in exergonic reactions, you saw in the exergonic reaction that there is a net loss. There is a net loss of free energy. So the products have less free energy than was present in the reactants. And the reaction proceeds spontaneously. And you can see this class on page 153. Getting back to diffusion, which was, of course, on your prior test, that random moving particles that diffuse down their concentration gradient. Well, as it happens, free energy decreases as entropy increases. So that provides the energy for this apparently directed process known as diffusion is the concentration gradient itself. That's how those particles move from that higher to lower concentration until equilibrium is reached. So when I say it this way, a cell must expend energy to produce. I repeat, a cell must expend energy to produce the concentration gradient. Keep that in mind. So, having stated that, because work is being done to produce this order, the concentration gradient class is a form of potential. It's a form of potential energy. The concentration gradient is a form of potential energy. So as the particles move about randomly, the gradient becomes degraded, and thus free energy decreases and entropy increases. So let's pair this with where we're going. So where we're about to go is here shortly to respiration and photosynthesis. So it will be that potential energy stored in the concentration gradient of hydrogen ions. And I'll just draw right there. That is a hydrogen ion. So as that gradient is built of those hydrogen ions, of course, it will then be transferred or transformed into chemical energy in the form of adenosine triphosphate, known as ATP. And that happens as those hydrogen ions will begin to pass through the membrane, going down the concentration gradient. So this concept, class, which I'll just give it to you now, will be known as chemiosmosis. We haven't quite gotten there yet, but that's where we're going. That's where we're going. So with this, free energy does change in a chemical reaction, and it depends on the difference in bond energies between the reactants and products, and even the concentration class of both reactants and products. So let's continue on now. So in the event that the reactants have much greater free energy than the products, most of the reactants are going to be converted to class II products, or vice versa. So with endergonic reactions, to keep this in mind, it's just a bit different in that they're sometimes, or at least they are coupled, with those exergonic reactions. And when I say coupled reactions, I'm referring to those thermodynamically favored exergonic reactions that provide the energy to drive those thermodynamically unfavorable endergonic reactions. So to make this make a lot more sense, I'm going to bring in ATP with this. So in a living cell, what I'll say here is that the exergonic reaction often involves the breakdown of ATP. So let's get now to ATP. So you'll find this on page 155. This is section 7.4. Let us begin. So here in section 7.4, it is all about adenosine triphosphate. In all living cells, energy is temporarily packaged within a remarkable chemical compound known as adenosine triphosphate. This is what holds readily available energy for very short periods of time. So it states that we may think of ATP as being the energy currency of the cell. When you work to earn money, you say that your energy is symbolically stored in the money you earn. Keep that in mind. The energy the cell requires for immediate use is temporarily stored in ATP, just like cash. For those that use cash, and I hope you all still use cash. So when you earn extra money, you may deposit it in the bank. Hey, your cells do the very same thing, in a way that a cell may deposit energy in the chemical bonds of, I'll say, starch or glycogen, and even lipids. And particularly classes from animals, I would say in glycogen likely. And that will be stored in our liver and skeletal muscle. So with this, just as you dare not make less money than you spend, the cell must avoid energy bankruptcy as well. In other words, that would cause the cell to die. So you probably do not keep much money you earn very long. And the cell continuously spends its currency, also known as ATP, which must be replaced to class immediately. So ATP is a nucleotide consisting of three main parts. Those parts, class, are as follows. So we have our pentose sugar, and that looks quite ugly. Let me draw this again. So I say again, we have our pentose sugar, much better the second time around. And along with it, I'll draw here our adenine. So I'll put adenine right here. Connect it there with our porphyrin rings. So 2, 3. And then I'll go there, and go here, and there. And then we have our 3 terminal phosphate groups. So I'll go here, there, and I'll draw these pretty quickly. So there will be one here, there would be one there, and there will be one there, and they're connected just like such. Now, I drew this rather quickly for the sake of time. So more or less, this class is adenosine triphosphate, as it is. So with this, ATP is that nucleotide containing adenine, a nitrogen-containing organic base, as you see there now, the adenine, along with it class 3 phosphate groups. And they are identified class by way of the phosphorus atoms surrounded by oxygen atoms. Notice the phosphate groups are bonded to the end of the molecule in a series, such as you would see 3 cars behind a locomotive, like the cars of a train. They can be attached and detached. So with this, hydrolysis of ATP yields ADP, adenosine diphosphate. So we, of course, would have those 2 phosphates class attached, and of course we're left over with, I guess I'll call it, let me just draw it a different way. Give me just a moment. I'll draw this a different way. With this inorganic phosphate here. So we now, instead of having ATP, we have class ADP. We have, of course, adenosine diphosphate. And then, of course, we have that inorganic phosphate there. Next up. So as ATP donates energy, what you should, I say, keep in mind is that it's an intermediate reaction that's there for the formation of sucrose. That in this case, it involves a phosphorylation reaction. And I can understand this is a new term, but let's get to that new term and make it make sense. So the phosphate group will be transferred to the glucose to form glucose P, or that glucose phosphate. So let's make it happen. So we have glucose and ATP. This is where I am, class, if you're wondering. We have glucose and ATP. I'm going to add a bit of color because I think this is a bit bland. There we go. So we have these 2 reactants and, of course, products as such. So what's happened is glucose became phosphorylated. Look closely, class. There was the addition of that phosphate, and what was once ATP is now ADP. And with this, the very same can be shown by saying we have this glucose phosphate plus sucrose to yield, plus fructose to then class synthesize sucrose and this here inorganic phosphate. Donation of energy. So just keep in mind that ATP links exergonic and endergonic reactions. So those exergonic reactions, of course, they liberate and release energy. They liberate and release energy. That's what they do. And there you go, that release energy then drives endergonic reactions. So with that being the case, I'll now move on to the typical cell and how it maintains a high ratio of ATP to ADP. So the typical cell contains 10 more, I repeat, more than 10 ATP molecules for every ADP molecule. So the high levels of ATP makes its hydrolysis reaction more strongly exergonic, and more able to drive those coupled endergonic reactions. And just keep in mind that our cells, your cells, the cells cannot store large quantities. They cannot store large quantities of ATP, and ATP is constantly going to be used and replaced. And as I mentioned moments ago, if energy is going to be stored, it's going to be stored class in the form of glycogen, or at least in the form of starch, or even class in the form of some lipid within us. So next is section 7.5, and we're getting closer to the end with energy transfer and redox reactions. So this goes back to chapter 2 class with basic chemistry, in which you were taught that energy is transferred through the transfer of electrons from one substance to another. So it was referred to as being oxidation and reduction. So a substance that is being oxidized is actually losing those things on its electrons, and the substance class being reduced or going through reduction class is gaining electrons. So having stated that, I hope that was a good review from what we've done. So getting to this, just keep in mind that you've seen that cells transfer energy through the transfer of the phosphate group from ATP, though energy is also transferred through the transfer of electrons. So redox reactions often occur in a series of electron transfers. I say in a series of electron transfers, i.e. respiration class, photosynthesis, and many other chemical processes. This is what's occurring, meaning those redox reactions. So redox reactions in cells usually involve the transfer of a hydrogen atom. So there we have it. So an electron, along with its energy, is transferred to an electron acceptor molecule, such as the nicotinamide adenine dinucleotide. Yes, that's NAD. You hear me say NAD instead of saying nicotinamide adenine dinucleotide, because that would be a mouthful to say every time. So, what happens, class, is NAD plus, grab a different color. What happens, class, is NAD plus becomes reduced, and it's reduced, class, to what is known as NADH. So with that, there was just, class, a transfer of energy. Now, with that being the case, let's continue on, because you will see this again, meaning it's I guess I'll put it this way. NAD class, meaning it's called nicotinamide adenine dinucleotide. It's one of the most common acceptor molecules, class, in cellular processes. You'll hear me say it over and over again. So as it becomes reduced, class, it temporarily stores large amounts of free energy. And I'm going to show you this again in just, I'll show you now. So if you look closely here, we have these two nucleotides, of course, linked together, class, by the phosphate backbone here. So as I show you this, just please keep in mind that NAD, of course, in its oxidized state, is just NAD. However, class, as it becomes reduced, it becomes NADH. So as it becomes NADH, it now, class, has that energy. So the way I put this is that NAD consists of those two nucleotides. I guess I'll say this one and that one. So one with adenine, and of course one with nicotinamide, which I think you all can now see. Nicotinamide, and of course the other class with adenine. So the oxidized form of the nicotinamide ring is there and becomes reduced class NADH, which I just mentioned. So it's all by the transfer class of 2 electrons and 1 proton from another organic compound. So there you have it, class. So here just keep in mind that an electron progressively loses free energy as it's transferred from one electron acceptor to another. So in respiration class, NADH transfers electrons to another molecule. So keep in mind that that energy is then transferred through a series of reactions that result class in the formation of adenosine phosphate. So NADP is not involved, particularly class in ATP synthesis. However, electrons of NADPH are used to provide energy for photosynthesis. So I say these two because we'll get class to NAD, and we'll think about NAD, but we'll also then get class to NADPH in photosynthesis. Meaning I'm saying this because they're involved class in two different processes. The first of which class being respiration, and then the latter of the two being NADPH class being involved in photosynthesis. So the next subclass is flavin adenine dinucleotide. FAD is what I'll call it. I'll say again, flavin adenine dinucleotide. I'll just call it class FAD. So FAD is a nucleotide, and it also accepts hydrogen atoms and their electrons. So, as I mentioned before, class, redox reactions, what happens is that FAD will become reduced to FADH2. Thereafter, there'll be cytochromes. So, cytochromes are proteins that contain iron, such as, I guess, one of the more well-known ones being known as cytochrome C. So what then happens is the iron component here accepts electrons from the hydrogen atoms and then transfers the electrons to some other compound. So I hope that you can see, class, that it's important to know that these electron carriers are carrying, class, everything we need as far as energy to, of course, synthesize energy in the form of ATP, or even, class, in the case of what I mentioned, NADH, power class, photosynthesis. And that's NADPH. Now to enzymes, and let's finish this chapter. So the principles of thermodynamics help us to predict whether a reaction can occur, but they tell us nothing about the speed of the reaction. You'll find me, class, at the bottom left-hand corner of page 158. The breakdown of glucose, for example, is an exergonic reaction, yet a glucose solution stays unchanged virtually indefinitely in a bottle if it is kept free of bacteria and molds and is not subject to high temperatures or strong acids or bases. Cells cannot wait for centuries for glucose to break down, nor can they use extreme conditions to cleave glucose molecules. Cells can then, of course, regulate the rates of chemical reactions with enzymes. Here we go. Enzymes, class, are known as biological catalysts that increase the speed of chemical reactions without, I repeat, without being consumed by the reaction. In other words, they can be used, class, over and over again, as long as, of course, the substrate is there. So although most enzymes are proteins, scientists have learned that some types of RNA molecules have catalytic activities as well. Cells require a steady release of energy, and they must regulate that release to meet the metabolic requirements. So, metabolic processes proceed by a series of steps that a molecule may go through as many 20 or 30 chemical transformations before it reaches its final state. For instance, one thing that I've mentioned to you all, class, is that catalytic ability of some enzymes is truly impressive. And for instance, with hydrogen peroxide, this H2O2, I gave you the equation once with 2H2O2. But what hydrogen peroxide does, class, is break down the extremely slowly if the reaction is uncatalyzed. But a single molecule, known as catalase, comes in, class, and brings about the decomposition of 40 million molecules of hydrogen peroxide per second. And I can hardly wait to show you all this in person in lab. So catalase has the highest catalytic rate known for any enzyme. So it protects cells by destroying class that toxic hydrogen peroxide. Yes, it's poisonous class, and it's produced as a byproduct of some cell reactions. So the Bombardier beetle uses the enzyme catalase as a defense mechanism. So it's pretty amazing how this works. So to keep you there, class, the energy of activation. This is that energy class needed, or at least required, to break those existing bonds and begin the reaction. So what I like to say, class, is that enzymes lower. I like to say that enzymes, they lower the energy of activation. And you may hear others, class, call it activation energy. You may hear others call it activation energy. You may. So, having done this, we'll keep on moving here, class. We'll keep on moving here. So look closely. All reactions have the required energy of activation. So if you look closely, this is pretty much what I've drawn for you prior to now, when we had gotten to proteins and enzymes, briefly in, I think it was chapter 3. So as you see it, please tell me what you all see here, indicated, class, by the reaction there in red. What do you all see? And I guess to ask a better question, what can you all say about the progress of this reaction in red compared to the progress, class, of that reaction in gray? Hope you all can tell, class, that the reaction occurring, class, without an enzyme, A, required, class, more, I repeat, so much more activation energy compared to, class, that very same chemical reaction occurring, class, with the addition of an enzyme requiring that much activation energy, shown there, class, in purple. So this is a stark example, a perfect example, showing how not only, class, do enzymes lower the activation energy, but they also, class, speed up that rate of reaction. This enzyme proceeded taking that long, that reaction proceeded taking that long, as opposed to this reaction proceeding, class, taking that long. And for some reason, class, it made my line look longer than life, if I can say it that way. There it is. Continuing now. So what happens, class, is that enzymes work by forming what is known as the enzyme substrate complex. And to help you with it, I'll be coarse in my illustration of this, class. I'll be very coarse. I hope it makes a lot of sense. Let's just call this the E for enzyme. And now I'll just change the color up just a bit. And I'll go with, let's go to yellow piece. So that's the yellow piece. It's about to come, actually. But that's the enzyme. So enzyme, class, plus substrate, which I have now, class, here in yellow. Yellow, class, is substrate or substrates. We have now, class, created what is known as the enzyme substrate complex. With this, class, keep it in mind that as the enzyme and substrate have now, class, formed that enzyme substrate complex, well, just keep in mind that this, of course, happened to, meaning the substrate, happened to bind to the enzyme at what is known the active site. So I'll draw, class, the coarse depiction of an enzyme here, yet again. And as I draw it there, I'll just put it as being that the active site is indicated here, class, by my purple arrow. And I say again, that is the active site, S-I-T-E. So as you see it, The active site is the place at which the substrate binds to the enzyme. So, having put it in such a way, I hope this is now something that you can understand a whole lot better. I'm not done. So, what then happens, of course, is that the active site of some enzymes are grooves or cavities. As you see here, the little cavity looks like almost like a square. So, the active sites of most enzymes are located close to the surface. So during the course of the reaction, the substrate molecules occupying these sites are brought close together to react with one another. So I'll just say that the shape of the enzyme is not exactly complementary that of the substrate. In other words, the binding site of the substrate to the enzyme molecule causes a change known as the induced fit. So when I speak to this induced fit, it's in the shape of the enzyme. It's just usually that the shape of the substrate also changes just slightly that may distort its chemical bonds. And this, of course, just ensures that it's going to be this enzyme class to bind to that substrate. Because it's not just any enzyme that will bind class to any substrate. It's highly specific. And well, let's just get to the specificity of enzymes now. So, enzymes, as I mentioned, class by the active sites, and they have that induced fit, causing that bit of a shape change. Well, what I'm getting to, class, about the specificity of enzymes is that lipase splits ester linkages in many fats. Then again, we may not think about lipase every day of our lives. Let's go back here. So, in this case, let's call this enzyme lactase. Go back with me, class. Please don't forget that many enzymes have the suffix ase. So there you go. That's lactase, class. There. And of course, I say even there. So the substrate class for lactase, you all know where I'm going, is in fact class what? Tose. Thank you kindly. So we call this class in yellow lactose. Lactase, the enzyme, binds with class lactose to create class that enzyme substrate product. So of course, upon the reaction class occurring, lactose class is broken down into its two component parts known as glucose and galactose. In other words, class, those are the two monosaccharides that make up the disaccharide, lactose. And this all goes back to, class, the specificity of enzymes. In other words, due to that shape of an active site and its relationship of the substrate, some are specific, class, only to a certain chemical bond. And of course, with this, that's how they remain specific to, of course, this enzyme binding to that substrate. So many enzymes require cofactors. And sometimes it's called an apoenzyme and a cofactor. So neither alone can do anything class kinetic. In other words, they cannot function separately. They must class function together, meaning only when the two are combined. So cofactors may be a specific metal ion, such as zinc, copper, or iron. Keep those in mind. So, having gotten this far, just keep in mind that an organic non-polypeptide compound that binds to an apoenzyme and serves as a cofactor. So with this, I'm referring to, of course, most of them being those scary molecules. Saying, what do you mean? Well, NADH, thanks a lot, NADPH, and even class FADH2, which of course serve to transfer electrons. The electron carriers are called. ATP transfers phosphate groups, and coenzyme A, which we'll get to, class, here shortly, known as, we'll get to it later, but it transfers groups derived from organic acids. So most vitamins are coenzymes or components, class, of coenzymes. So this is so amazing what we're getting, class, and if it doesn't make much sense yet, please ask me questions, and of course, I say, keep listening. So now I'm here. Enzymes are most effective at their optimum. So optimal conditions, class, are those, I guess you say, how enzymes generally work, meaning they work best under those narrowly defined conditions, such as the appropriate pH, such as the appropriate temperature, and even the appropriate class ion concentration. In any class, deviation from such can adversely affect class the enzyme's activity. And I'll help you out. Can adversely affect class the enzyme's structure, and of course I just mentioned its function. So each enzyme has an optimal temperature. And most enzymes have that optimal temperature at which the rate of reaction is fastest. For humans class is about 35 to 40 degrees Celsius. And to keep it in mind, when I say 35 to 40 degrees Celsius, if you were to look in your text, of course, to go from Celsius Fahrenheit, 35 degrees Celsius class is an approximate 95 degrees Fahrenheit. And to go to 40 degrees Celsius is an exact 104 degrees Fahrenheit. At least you now know. So as I'm here, I guess I'll continue where we are. Look closely. Most human enzymes have that optimum class there, as opposed to class enzymes of heat-tolerant bacteria have their optimum temperature class there. And I did not mean to put dotted lines there. Excuse me. There we are. So keep that in mind as we get to, of course, what's next. Well, let's stop at Archaebacteria, the Archaea. These heat-tolerant bacteria have enzymes that allow them to survive extreme temperatures, such as, of course, what you're seeing now, out of Yellowstone. Wherever you all go, please take photos and email those to me. I like pictures, and I cannot wait to visit Yellowstone. Back to work. So, as far as pH is concerned, it states that, of course, if in fact, class, those enzymatic controlled reactions occur, and I'll just go backwards for a moment, and temperature class increases beyond that optimum condition, it can very well, class, cause the protein to rapidly denature. It could cause that protein to rapidly denature. I say in the case of human enzymes getting too warm, or of course being class far too cool. And by denaturing class, I mean that protein, that enzyme class will lose both its structure and its function. So the molecular conformation, that 3D shape of the protein, becomes altered as the hydrogen bonds that are responsible class for its secondary structure, its tertiary structure, and quaternary structure, they become broken. And this is an irreversible change. And of course, the activity of that enzyme class is gone. It will not function. Back to what I was going class pH. I'm changing my color from green. For whatever reason, this green just doesn't look good. So most organisms, class, are killed with, of course, short exposure to high temperatures, and their enzymes are denatured. So please watch out. And the same is the case, class, when, of course, certain organisms get too cool. And I didn't mention the temperature yet, so I kind of went briefly through here. Class, the temperatures here that you're seeing, class. And look closely, and do you see there in the, I guess I'll say, background? The temperatures here, class, can get upwards of 100 degrees Celsius, class. That's 212 degrees. So if that's not hot, I don't know what hot is in these brilliant colors we see, class, from those terraces of the hot springs of Yellowstone. All right, I promise I'll show you class at pH. So most enzymes, class, are active only over a narrow pH range, and of course have that optimal pH at which the rate of the reaction is fastest. So the optimal pH for most human enzymes is between 6 and 8. Keep in mind, class, that we, as humans, class, have an amazing mechanism called the buffers, class, in our buffer systems to ensure, class, that the pH happens to not change in the body. It remains constant within that narrow limit. For instance, our blood pH class, it remains 7.35 to 7.45, thanks to, of course, our amazing buffer systems. And that's going to be blood pH at homeostasis for us. So let's get down to where we're going. Let's begin to the left here, class, with pepsin. So I don't know what you know about pepsin, class, but pepsin is what I know as an enzyme that digests proteins. Pepsin, class, is found within the stomach. So it digests proteins, secreted, class, by cells that line our stomach. So it works, class, in a very, very what type of environment? I hope you say a very acidic environment, class. So yes, that might kind of buck the trend, not being at this approximate 68 as far as pH is concerned, but its optimal condition, class, is in that very acidic stomach. And of course, it breaks down proteins there well. As opposed to trypsin, as you all now see, it's also, class, a protein splitting enzyme that's, in this case, secreted, class, by our pancreas, and we find this, class, in the small intestine. Something, class, that will help you is look at its optimal temperature. I'm sorry, its optimal pH, excuse me. The optimal pH here, class, is at approximately 8, where, of course, things are much more alkaline, class, in the small intestine. So the activity of an enzyme may be markedly changed just by altering, class, the pH, which in turn alters the electrical charges on that enzyme, which of course affect the ionic bonds that contribute, class, to both the tertiary and quaternary structures, thereby, class, changing the protein's conformation and activity, i.e., class, the protein becomes denatured, losing, class, both structure and function. So please keep this in mind, class. This is, I say, quite important to know. For instance, if trypsin, class, were to get into the stomach, would it function? I hope you say no. And the same, class, will be the case if, of course, pepsin, class, got into what is known as the small intestine. Metabolic pathways. And let's finish this chapter up. So with metabolic pathways, it's just a product of one enzyme, class. One enzyme controlled reaction, of course, serves as that substrate for that next series of reaction. So if you happen to remove an intermediate or a final product, of course, it could most definitely cease to cause that metabolic pathway to continue. So enzymes combine through one or more multi-enzyme complex. And what I'm saying about that is, it just may be that we have enzyme 1, we have enzyme 1, and of course, enzyme 2. I guess I'll go as far as enzyme 3. So with those 3, of course, there are substrates. So enzyme 1 has a substrate, enzyme 2 has a substrate, and so on and so forth. So in the event, and those should have been in a different color, in the event, class, that either the substrate, class, or the enzyme were to be missing, it would stop, class, the entire metabolic pathway, no matter, class, where it occurred. So without substrate and enzyme, the pathway, class, would cease, no matter where it is. So genes, class, also regulate enzymatic activity, meaning it's one gene that codes, class, for one protein. For instance, what I'm getting at here, class, is a specific gene is what directs the synthesis of each type of enzyme. So the gene may be switched on by a signal from a hormone or other signal molecule. In the very same way, the amounts of enzymes also influence the overall rate. So depending upon, class, whether you need to synthesize more or less of that enzyme, it's all controlled, class, by gene expression. So as you can see here, class, as the concentration of the enzyme, class, increases, so does the rate of the reaction here. However, as you can see on the converse, class, as the concentration, class, of the substrate increases, you see here, class, that the rate of reaction class appears to level off. And I'm saying this to ensure that you see that the rate of the reaction is measured class at different substrate concentrations and enzyme concentrations, and even the temperature and pH are constant. So what I'm stating is that as the substrate concentration is low, the reaction rate class, of course, is the approximate low as well. However, as that substrate concentration class increases, it does not class change. I repeat, it does not change the rate class of the reaction. I don't know what happened to my arrow, but it does not class change the rate of the reaction, as you can see here. And it's all because the enzyme has become saturated with the substrate, as opposed to what I showed you class firstly here in figure 714a on page 162, that of course, with an excess amount of substrate, of course, the rate of the enzyme is directly proportional to the enzyme concentration. So now the product of one enzyme reaction may control the activity of another enzyme in this sequence of reactions. So just keep in mind that when the concentration of the product is low, the sequence of the reaction proceeds rapidly. However, when the concentration of the product is very high class, the reaction will then, of course, stop to ensure class that it is not far too much. And this kind of gets us, of course, into what we're going with feedback inhibition, meaning the enzyme regulation happens in which the formation of that product will feed back class to inhibit that early reaction in the sequence to, of course, stop the production class of that product. Now, finally, class, we get to inhibition. And not feedback inhibition, but we're going to get to allosteric sites. So some enzymes have a receptor site called an allosteric site. And what happens is the allosteric site is just some region of the enzyme molecule other than the active site. So when the enzyme has a substance that binds to its allosteric site, that changes the enzyme's shape and modifies its activity. In other words, allosteric inhibition is what occurs, meaning that change in state inactivates the enzyme. That way, of course, the substrate cannot bind to the enzyme, and it's all due to allosteric inhibition. For instance, some of which can be called those cyclic AMP, meaning those CAMP class dependent protein kinases. So the allosteric activators, they result in that functional active site. In other words, if you look closely, class, in figure 716, 716b shows you, class, the active form of the enzyme based on, class, the removal of that allosteric inhibitor, which of course activates the enzyme. So there you have it. Looking closely here, I guess I'll call this, what color is it? Red, okay? So we have cyclic AMP, that regulator inhibitor. So at the allosteric site, we have, of course, an active site that is inhibited, and it's all based on this inhibitor molecule being bound class to that allosteric site. In B, we have cyclic AMP. Cyclic AMP comes, class, as I just mentioned, to remove that allosteric inhibition class and activate the enzyme, allowing the substrates to bind, giving us, class, again, that enzyme substrate complex. So there you have it. So next up, class, I'll get to briefly, class, competitive inhibition. So what we're seeing here, class, in A as an apple, and I'll go back to this black piece. What we're seeing here, class, in A is that we have this competitive inhibitor binding, class, to the force active site. And I say it's a competitive inhibitor. That's a long word, huh? Competitive inhibitor. Because it competes, class, for the active site. So now that that purple inhibitor is bound to the active site, those substrates class, they cannot bind to the protein. They cannot bind to the enzyme. And it's all because of competitive inhibition. And of course, if it happens class to be a non-competitive inhibitor, that just means the inhibitor class binds to a site other than the active site. So when I got to just previously class, allosteric sites here, moments ago, that was an example class of non-competitive inhibition. That was a form of non-competitive inhibition. So lastly will be the irreversible inhibition. And with this, it just permanently inactivates or destroys the enzyme. And many poisons do this, such as heavy metals that you I hope know about, class, such as lead, mercury, nerve gas, and even cyanide. Those class are definite poisons. Those are definite killers. Please watch out for those. So death results class from cyanide poisoning because cytochrome oxidase irreversibly inhibits and no longer transfers electrons from its substrate to oxygen. So, in other words, class, as those electrons move down the electron transport chain, oxygen class would normally be that final electron acceptor. And of course, no longer is that the case. No longer is that the case. So this class has been your instructor, Scholar Huff. This has been Chapter 7. Prepare well, study well, and take notes. Thank you all for listening.